Okay, chapter 3 is about microscopy, observing microorganisms through a microscope. In here we have a nice picture of Helicobacter pylori. This is color enhanced with a scanning electron microscope. This Helicobacter pylori, it's a gram-negative bacteria that causes cancer, gastric cancer, cancer in the stomach. It's actually the only microorganism, bacteria, that causes human cancer. The rest are caused by viruses. And this has a rod shape and it has an extension that it's a flagella. And this colonizes the pyloric region in our stomach, and it can cause initially a inflammation to what we call gastritis, and then as time progresses, it can cause an ulcer. And that ulcer ultimately can become malignant, and the person will develop cancer because these bacteria in itself will basically inactivate tumor suppressor genes like p53. The problem in here is chronicity of the infection. Someone can have an acute infection and the person basically can be treated really easily with a cocktail of antibiotics and some antacids. But if the person just plainly go on with this infection, chances are that the person will develop gastric cancer plus other factors like smoking, eating a lot of salty foods, etc. Now, the importance of this is that we have so sophisticated microscopes that for sure, unequivocally, if you use, for instance, this type of microscope to see these bacteria, I mean, no one can tell anything different, okay? So, anyway, so here we have this image showing you the different types of microscopes that we can use to see a specific objects at a particular length. So we have our eyes, of course, that can see objects up to something that is 100 micrometers. I mean, if you're young, you have good vision, you can see something up to 100 micrometers. More than likely will be something greater or equal to 200 micrometers, and we will see the units in a bit. But for instance, we have here thick in the skin of a person, for instance. You can see it without, if you don't wear lenses, of course, you can see it without lenses. Now, red blood cells, they can be seen with a light microscope that can see objects between 200 nanometers and 10 millimeters. Or we can see it with a scanning electron microscope between 10 that can see objects between 10 nanometers up to 1 millimeter, which is here. So this scale shown in this axis, it shows objects from 1 meter up to 10 picometers, which is a very small unit. So the smaller units are on the right, the larger units are on your left. And by the way, when we observe objects in microscopy, we use, of course, the metric system, which includes the unit of length, which is 1 meter. So then this is the range for light microscopy, not a light electron, light microscopy between 200 to 10 millimeters, the scanning electron microscope between 10 nanometers, which is here, and 1 millimeter, TEM, transmission electron microscope, between 10 picometers and 100 micrometers, atomic force microscope between 0.1 nanometers and 10 nanometers. So here are the different views of the different microscopes, and of course there are abbreviations in this box. So like microscope, you can see an erythrocyte, a red blood cell. And if you see here, again, we have these bar scales that if you extrapolate, it gives you basically the thickness of this erythrocyte. So an erythrocyte can be, I guess, based on this, up to 6 micrometers. It's between 6 and 8, okay? And then here we have E. coli bacteria that if you extrapolate this bar scale, I mean, by length, it will be more or less like 2.5 micrometers, and by width, maybe a little bit less than 1 micrometer. With TEM, you have here visualization of an actual virus that can infect E. coli, T. even, bacteriophage. So this one looks like a little alien ship. It has this pin that can connect into the surface of this bacteria and can inject the DNA to infect this bacteria. So this is the surface of the bacteria, and this basically, if you measure the basically thickness from the head, with this is the head of the virus, it can be basically up to 120 nanometers. And then this is basically double helix DNA as seen with an atomic force microscope, and typically the thickness of this DNA when it's starting to supercoil, it is 10 nanometers. Okay, when it's not relaxed, the molecule. So in this next part, we're going to list the units used to measure microorganisms, and we're going to do 1 or 2 calculations. So microorganisms, for the most part, the ones that are not visible, we will be measuring them in micrometers. Greek letter mu, this one, it is basically used to denote before letter M, lowercase, to denote micrometers. And also for those microorganisms that are small, and I mean viruses, not microorganisms, but viruses can be measured in nanometers. And in this case, you use NM as an abbreviation. And then the equivalents. So in 1 meter, we have 1 million micrometers. 1 micrometer is equivalent to 10 to the minus 6 meters. And we have 1,000 millimeters in 1 meter. Now, 1 nanometer equals to 1 billionth of a meter, or 10 to the minus 9. And we have 1 million nanometers in 1 millimeter, or 10 to the minus 6. So, in 1 millimeter, we have 1,000 micrometers. And in 1 micrometer, we have 1,000 nanometers. So you can say that 10 to the minus 3 or 0.001 micrometers equals 1 nanometer. And again, we have 1 million nanometers in 1 micrometer. Sorry, in 1 millimeter. Or 1,000 in 1 micrometer. So, how many nanometers do we have in 1 millimeter? In 10 millimeters. Let me go back to that. So if you see there, we have 1 nanometer, which is equivalent to 10 to the minus 9 meters. So basically, these will be the same as to 10 to the minus 6 millimeters. So in 1 meter, we have so this is the number of zeros, right? Nanometers. And then these nanometers, so 1 nanometer equals 10 to the minus 6 millimeters. So what will be the number that I will put here according to this? How many zeros after the 1? How many? 6. So how many nanometers, how many nanometers we have in one millimeter? I wrote that incorrectly. 1 million, right? Because 1 nanometer is 1 millionth. This is the word. When it's the minus 6, that means that it is 0.000000, and then should I continue with the 0 and a 1 or just the 1? Just the 1, right? Because when this 1 is minus, that means that you will be moving to the left, that dot, okay? The point. So then, if we have in 1 millimeter, we have 1 million nanometers, then what you will do here? To get the number of nanometers in 10 millimeters. Where?
It's the 1 million.
So if I add another 0 here, let me do it here. So if I add another 0 here, then I have to change this, and then how many we have? 10 million, right? So 10 millimeters equals 10 million. I mean, another way is just multiply this times 10. Because let's say that you have, in this case, instead of 10, like let's say that you have 15, or you cannot add 1.00, right? So in this case, you can multiply 15 times this, and then you will get your 15 million. Okay. So now let's look at the different types of microscopes and how they produce the image. And then we're going to see what is magnification and resolution. So we have the simplest type of microscope, which is made out of a single lens, and is similar to a magnifying glass, but much better, higher magnification lens. Actually, if you wear glasses, basically that's a microscope, okay? And then you remember this is the replica of what Antoine van Leeuwenhoek developed to see smaller microorganisms. I think he could magnify up to 400 times or so. And then the common type of microscope that it is used in academic settings, it is the light microscopes. They have different categories, including compound light microscope, which is the ones that we use in the microlab, dark field microscopes, phase contrast, DIC, differential interference, fluorescence, microscopy, and confocal. For compound microscope, it is called compound because you have a series of lenses that can magnify the initial magnification. In this case, we will have these objective lenses that increases the image size up to a particular number or size depending on what is the objective lens. So it can range between 4 or 5 up to 100 initial magnification. And then the next, the ocular lens, which typically in the microscopes that we use is 10 times, it magnifies 10 times more. Sometimes there can be some ocular lenses of 20 times. But the application, it will vary, I guess, depending on what lab you will be working at. So then these objective lenses are hauled into the tube of these objective lenses at the tip, and they can become smaller, and they are hauled into this nose piece. So whenever someone is using a microscope, it's supposed to change the objective lenses by grabbing it from the nose piece. And then here is the arm of the microscope. This is the base, and in the base we have the light source or illuminator. We typically have also the light switch, and then we have 2 knobs, the fine focus knob and the coarse focus knob. And then we have in the front of the arm the stage that holds the slide into position, and then we have below the stage this condenser lens that focuses a little bit more the rays of light that come from the illuminator into the specimen. That's why it's called condenser, because it condenses, brings to a single line. And then microscopes as well, they have a diaphragm that controls the amount of light that goes through the condenser. And then we have these knobs or screws that moves the stage front and back and side to side so that you can scan your sample until you find whatever you need to see. And then in this part that is called the body tube, you have inside some prisms that can bring the light, specifically directing it into the ocular lens, which basically has the lens here, and then you can have either some pointers attached into these lenses so that you can basically point to that specific object. Some of them, they might have grids that help you to calculate the size specifically of the specimen. So then this is the area that you use, or the structure that you use to see into the microscope, into your image. And then in order to obtain the total magnification, you all have to multiply the times the objective lens is magnifying the image times the ocular lenses magnifying the image. So maximum magnification that we can obtain is 1,000 times. And this is the pathway of light of the compound light microscope. So everything starts here in the illuminator with several rays of light going parallel towards this condenser lens that condenses specifically into the level, the light into the level of the specimen that is found typically in a glass slide. And then the light will be refracted going into this objective lens that makes this basically rays of light going into this prism inside the body tube that can have that physical property of bringing specifically the path of light towards the line of vision in the ocular lens. So microscopes, they have something that is called the resolution or resolving power, which basically it is the property of how sharp the image is going to be depending on the quality of the microscope, specifically the lenses. So resolving power properly is defined as the ability of the lenses to distinguish between 2 points. And then it can be measured in nanometers. So for instance, a microscope with a resolving power of 0.4 nanometers means that it's able to see the distance between 2 objects of 0.04. That's the minimum distance. The microscopes have, in the case of a microscope, it has a resolution limit of 0.2 micrometers. So in the case of these light microscopes, yeah, you can see the objects clear, but of course not as clear as more sophisticated types of microscopes. And then sometimes you can have this magnification up to 1,500 times. Something that also affects the quality of the image is what we call the refractive index. Refractive index is the measure of how much the light is going to bend as it's passing through a medium. So you can decrease this refraction, and then you can have the light, instead of bending away from the pathway of light, it can go more parallel by reducing this refractive index. How can you reduce it? Well, by adding, for instance, oil between your sample and the objective lens. That way you can capture as much light. And this oil will have similar refractive index as the glass light that you're using. Anytime that a wave of light passes through a physical object, that light is going to tend to refract or bend away. So then when you lose light, as it's passing through this objective lens, you lose resolution. So you increase resolution by adding some medium. So here is a magnification of the area between the condenser lens and the objective lens. Of course, in the middle we have the glass slide that is held by the clips on the stage, and then here we have, of course, the stage that is not shown. But basically, the light source sends these lenses, these rays of light that goes through these condenser lenses. Condenser lenses is basically focusing into a single point, focal point, which it's supposed to coincide to where the specimen is located in the glass slide. And then these ones, of course, not perfectly will go straight. It tends to go a little bit to the sides, refract, but if you don't have anything between the objective lens and the glass slide, you will lose a lot of rays of light that goes to the sides into the air. But that's when only with the oil immersion objective lens. That's when you use oil, and that oil is going to prevent so much of this refraction. So you will have more rays of light. I mean, in here they just show one, but you have more rays of light going through this objective lens, and you have a better resolution. I don't know if you have done this before, but if you put, for instance, a piece of paper with letters that are very tiny on the bottom of a glass, you will still see it tiny. But if you add a drop of water on top of that glass, the image is going to see, and of course it has to be held, not to going sideways. But if it's contained with that specific area, you will see a little bit larger that letter. Because you lose less light. So now for bright field illumination. So bright field microscopes, like the microscope that we use, basically it is a microscope that allows you to see a dark object against a light background. And that dark object is actually not going to be naturally dark unless, well, when it comes to tissue or actually bacterial cells, okay? Unless basically this microorganism naturally has pigments. But if it doesn't, then you have to stain it to increase contrast. If you try to see a specimen that is not naturally stained or, well, does not have natural pigments, with a microscope that is basically bright field, you won't see as much. So that's when you start staining this to increase the contrast so that you can see a dark object against a light background. So here we have this specimen, which is a protozoan paramecium. So this is a scene with this bright field, and in this type of microscopes, you have, of course, the illuminator that sends the light through this condenser lens that brings the light into the specimen, and then that light is picked up by the objective lens and the ocular lens. This specimen has natural refraction index that allows you to see part of these details in the specimen, but not all of them has this natural refraction index. So you don't see as much of these other specimens. But in here what you can see is this outline, which is called pellicle, and then you see a lot of extensions. So these are like pili that this microorganism uses to swim. It lives in aquatic environments. So here is an animation that we won't be able to see, but before we move into different other types of microscopes, so this answer this question. Through what lenses does the light pass in a compound microscope? Which lenses? What lenses? Condenser, and what else? Objective, and it passes through three ocular lenses. Then what does it mean when a microscope has a resolution of 0.2 nanometers?
It's the minimum distance to things can be measured.
So that's the minimum distance to see an object that is separated 2 nanometers, 0.2 nanometers apart. Do you think that will be a high resolution microscope? rather than 2 micrometers? Which one will be having more resolution? One that it has 2 micrometers that can see objects 2 micrometers apart, or one that is 0.2 nanometers apart? Yeah, this one has a high resolution. I took out my phone because if I compare my phone to your phones, yours has better resolution taking pictures. You can see the objects sharply. More sophisticated, less sophisticated. Compound microscope, scanning electron microscope. With your phones, you can even see what the people is thinking. Okay, now we're going to see other microscopes like dark field, phase contrast, DIC, fluorescence confocal, two-photon scanning, and we're going to compare each with bright field illumination, and we're going to see how electron microscopy is different from light microscopy, and examples of TEM, SEM, and scanning probe microscopy. So dark field is similar to bright field. It uses light as a source of illumination, but that light, it is blocked by diaphragms that exist in the condenser lens, by opaque disc. So that will block the middle layer of the pathway of light, and then an object that it cannot be stained because it can get damaged by the staining technique can be seen as clear against a dark background. That's why it's called dark field microscope. Typical example of a microorganism that it is used to, or well, that can be observed through these microscopes, it is Treponema pallidum. The bacteria, spiral bacteria, that basically causes syphilis. This bacteria is so slender and so fragile that if you try to stain it, it will be destroyed. So then you use this dark field microscopy. So here we have a Paramecium species, this protozoan, now seen with this type of microscope, dark field. So in this case, we have in the condenser lenses this opaque disc that blocks these rays of light that goes in the middle. So only light that gets refracted is being picked up, and then everything is going to be, and it's going to be refracted by the specimen, by the way. So then unreflected light, it is lost, and then you will have a dark field against a clear object. Phase contrast. So this one, it is used to see live specimens and the internal structures. So it is kind of a nice microscope because it can be used to study, for instance, the transit of nutrients or wastes within protozoans, for instance. And in this case, we have two sets of lights, direct rays and diffracted rays that forms an image. Some will be in phase, other ones out of phase. And then in this case, because with these two rays of light, you can see internal structures and differentiate from external structures. You don't need to fix these specimens and you don't need to stain them. So we have an annular diaphragm within this microscope, and then as the light goes, some of this light is going to be scattered by the specimen. In this case, you call it refraction, and diffraction. And then you have refracted or bent, naturally bent light. Both of them will be picked up by this part of the microscope, and then it will be brought into the plane of the ocular lens, and you will see a dark object, a dark field, and an object within this dark field that shows better resolution than the previous one. So DIC, differential interference, is similar to phase contrast, in which you use two beams of light, and you will have prisms that can split this light, and then it gives you almost a three-dimensional shape with color. And it will help you to see with better resolution these internal structures. So as you move, seeing the different images, you will see how these objects are seen better each time. With, I guess, one exception from my perspective. So here is what you can see with DIC. So this is paramecium. So you can see, for instance, vacuoles inside this. So it may have nutrients that this paramecium is having inside the cytoplasm. Maybe you will have some lysosomes with enzymes in here. And you can see these little, basically, pili around its plasma membrane. Fluorescence microscopy uses UV light, which is short wavelength. And these objects or specimens that you want to see with fluorescence need to be stained with a fluorochrome. A fluorochrome, it's a chemical or a molecule that can absorb UV light, and it can emit light at a greater wavelength. So it emits energy, and that can be seen as fluorescence. Depending on the fluorochrome that you use, it will be what color you will see. And you can use multiple fluorochromes, up to 3, I guess, to see different internal structures, including the nucleus, cytoskeleton, external structures like flagella, perhaps, pili. And these can give you differentiation then between the different structures. So some of these cells may stain, for instance, bright yellow or green or orange against a dark field. And then your book shows you a sample of Mycobacterium tuberculosis stained with this fluorochrome aramine O that makes the object look yellowish. Like in this case, this is Mycobacterium tuberculosis. One thing that in microbiology is important is that basically when you perform these fluorescence techniques in specimens that you believe are basically infectious either for animals or for humans, you use antibodies that are coated with these fluorochromes. And these antibodies specifically binds against receptors of these specimens. So there is less likely chances of having false positive results, because the antibodies bind to these receptors, and then you shine these fluorochromes with UV light, they emit the light, and then you can see the fluorescence. That means that your sample is actually what you believe it is. There is no discussion there. So fluorescence antibody technique is also known as immunofluorescence. So you don't necessarily need to use antibodies. Sometimes some objects naturally can be absorbing these fluorochromes, But when it comes to the surface of certain structures, including the plasma membrane, or even internally, like cytoskeleton or nucleus, and sometimes even DNA, I mean, it's very elegant what you can achieve with this technique. So you can see not only the surface, but you can see internal structures, and then you can see disruption of these structures as well when you mutate genes, for instance. So then provides a rapid, this immunofluorescence, rapid and specific detection in specimens. An example, this is Treponema pallidum, stained with immunofluorescence. This is one of these bacteria that you need to wear double gloves when you're handling, because this bacteria is highly motile in a corkscrew manner, and sometimes it can even penetrate through gloves. They go to your skin. Any questions? For confocal microscopy, the cells are stained as well with fluorescent dyes. So it's the same technique, you stain your specimen. But in this case, you can see different levels in the specimen. So you use same concept, emit UV light, send UV light into your specimen, which will send back a longer wavelength so that you can capture, or a shorter wavelength that you can capture with sensors in these microscopes that can be sent into a software in a computer, and it will give you this either single plane or stack image, and then you can reconstruct a three-dimensional shape, or you can see in a specific plane. It's like a CT scan. Like if they take a CT scan of your body and completely, I mean, you can see perhaps only the head, the neck, thorax, any part. Or you can stack it together and see the entire body. So here we have confocal microscopy of paramecium. In this case, the nucleus is not stained, but they have stained this cytoskeleton with green fluorescence. And then the pili with red fluorochromes. So it is the same paramecium, but now with a more sophisticated microscope. Okay, so it's time to stop.